Back again with Ken Hayworth. Now, it's interesting, there's been a lot of funding initiatives which seem quite large in the media recently. One of them has been the Obama Brain Project, or yeah, so that's one which is interesting. But there's been the IBM Blue Brain Project going on, which seems to be closely coupled with the European Brain Project or the Whole Brain Project. Is there any others that have been like massive funding for neuroscience recently?
Yes, I think the Allen Brain Institute
Mm.
should be considered in there definitely. I believe they got um, uh, a few hundred million
Wow.
additional funding for very interesting research into basically what they want to do is they want to look at a whole mouse brain while it is behaving under visual tasks. At least from what I've gleaned, they're still trying to figure out exactly what they're going to do. I mean, what they did with their initial round of funding from Paul Allen is they mapped all the genetic expression in a mouse brain. But now they are really targeting. It's basically the Obama Brain Initiative. Uh, it's already happening in private labs like the Allen Brain Institute, like the HHMI where I'm working, other institutes, and it's great to see a concerted push from the Obama administration in that direction. Hopefully, it'll get get funded.
Yeah. So, what are the differences between, like it seems as though it's great that there's lots of funding, but it seems as though there's funding for different approaches. Like the European, the Euro Brain Project, or the Whole Brain Project is seems to be different in approach to the Brain Initiative or the Obama Brain Project and the Ellen Institute stuff. So, do you want to discuss what the differences are and what the advantages are of each and disadvantages?
Yeah, that would be fine. I have to say the Obama Brain Initiative, and my boss actually just went to one of the kickoff meetings on that, trying to figure out exactly what is going to be done under that. Uh, so there's going to be a lot of different research uh, on mapping the structural connectivity of the brain, but also on a lot of activity. And there's really it's not yet determined at all, from what I've heard, what is actually going to be involved in that, which is great because there are a lot of different research directions that should be funded. Uh, it is absolutely the case that uh, that if we could record activity on a very large scale, all of the neurons in whole brain or all of the neurons in a key area of the brain, and do that with a very good temporal resolution, that would be fantastic. That would be phenomenal. It's likewise the case that if we could start mapping neural connectivity within brains, that would be fantastic. Putting those two together would be fantastic. And it looks like funding both of those technologies in parallel and getting them to work together is probably what is going to, hopefully, is that's going to look like. Because and I think that's very doable. I mean, the all of these the technologies that they're proposing, there are there are some far out ones like the molecular ticker tape and microbots that are attached. These things are kind of far out there, but there are other ways that also have been developed that actually are working on a smaller scale in labs that, with additional funding, could definitely be expanded and scaled up. Yeah.
Okay.
So the European project, I have to say, I I, uh, I, like most of the neuroscientists that I have talked to, uh, do not believe that there is going to be great breakthroughs coming out of the Human Brain Project, the European Human Brain Project. It's not that I think that the work that Henry Markram has done in terms of characterizing neurons in the cortex, all the types and all of their functional behaviors and the statistical connectivity between them from a functional perspective, all of that is fantastic work. I think that there are definitely things that will come from it, but the type of large-scale simulations, I don't see how those will work without knowing the actual circuitry that is underlying the actual brain. Because his simulations are based upon randomly connected circuits. Not randomly, I shouldn't say that, but they are certainly not based on an actual brain that has developed. They're based upon statistical connectivity. And he wrote a paper recently that said that's enough. I don't find that paper convincing. And most of the people I've talked to do not find that paper convincing. I think it's work that should go on, but should it get €1 billion, it's opportunity cost. There's a lot of other things that money should have gone to.
So I'd be more favorable of like draining the budget for creating new forms of lipstick marketing to fund the new approaches to neuroscience and draining the Human Brain Project or Henry Markram's project.
Yeah.
But I don't have an algorithm for sort of distributing costs that are currently sort of funding different aspects of neuroscience at all. No. I'd love to
Let me
know.
throw one thing in if I can.
Yep.
So, so, um, so the budgets that we're talking about are on the order of $1 billion,
Yeah, let's
right?
talk about budget.
Or multiple billions of
Hmm.
dollars. And to get any of these things to work on the scale that they eventually want to work at, they will eventually need to get a whole brain that is that can be imaged at the electron microscope level. They will eventually, they may not need to image all of it. They may not need to image all of it, but they will need to get a whole brain that you can look at different areas of that brain and trace long distance processes and then go in and do very high resolution imaging. And so it is a requirement for these to work, for these projects, and at least in my mind, for these projects to work, a chemical brain preservation technique, including plastic embedding, is so that it can be used, so that it can be imaged in an electron microscope, is an absolute requirement. Somebody should tell Obama this, somebody should tell Paul Allen this, somebody should tell the people that are funding this in Europe that brain preservation needs to be on their to-do list. Even if the only goals that they have are the goals that they've already said that they want to fund. And and that can be achieved with a tiny fraction of the resources that are being devoted to the other aspects of this problem. You know, buying the fastest supercomputer or developing molecular ticker tapes and things like that. The brain preservation aspect of this has to be solved, and it can be solved because it's a much more concrete problem. It can be solved for much quicker and much less expensively. And as an added benefit, you cure death. So why not just do it? Why not take 1% of that budget and just get the brain preservation aspect fixed? Now this is what, I think there is one researcher in the world that is working on this. There is one researcher in the world that is working on this part of the project that has to be solved if any of these are really going to, any of these large-scale ones are going to work. And that's Sean McCoolah in Germany, the competitor for the Brain Preservation Prize. And he has demonstrated, he hasn't passed all the requirements for the prize, but he has gotten very good results on a mouse brain that looks like he's got a technique that can, for a mouse, preserve a whole mouse brain at the level that you can then do electron microscopy on it. Mm-hmm. So, in my humble opinion, Obama, the European Union, and Paul Allen should get that guy on the phone and ask him how much he needs, how big the check should be. How big the check should be, because he's got the key to all of these Connectomics projects. He's got the key to understanding how the brain works.
Right.
And he's the only guy in the world that's working on it.
I'd like to interview him. What's his name again?
Sean McCoolah. And he's a very solid researcher, very solid. He's not talking about mind uploading or anything. He is just straight down the line. He is coming up with the best techniques that will get neuroanatomy to a level that is unheard of.
Right. And you've been following a lot of his work, and you've based some of your ideas about brain preservation on his work, or updated some of your ideas based on his work. What is it about his work? What research has he done that makes you think that he's worth listening to, or that Obama should fund his projects? What specifically, I mean,
Well, it's very concrete. I've seen the images. I've seen the images of his brains. I've cut brains that he is mouse brains that he's preserved, and looked at them under my electron microscope, and they look stunningly good. You can trace neural connectivity. You can see synapses. You can the structure of the neural circuitry is there, and he knows it's there because that is exactly what his mandate is. The laboratory that he's working on, the laboratory of Winfried Dink, they are all about mapping the circuitry of the mouse brain. They published a Nature paper a year or two ago that was a stunning reconstruction of a part of the mouse retina, along with functional recording, and showed that the function that was recorded in that piece of brain tissue could be predicted based upon the connectivity that was preserved and mapped by electron microscope in that part of the brain. It's basically the first uploading experiment, if you want to think of it that way. Mm-hmm. They've shown structure to function. There's a lot of other experiments that have also done that, so I'm not saying that. I'm just saying that they are a very serious group, and they know what they need out of a preserved mouse brain. They need to be able to have high quality electron micrograph images that can be traced from section to section to section, so that even the smallest neuronal processes can be followed for many millimeters. That is his mandate. And that is what he has developed. And he has been working on the chemistry of brain preservation, getting he's replaced some of the aldehydes with some other well-known aldehydes, but they get into the mouse brain. He's done some tricks with osmium, so that the osmium tetroxide that usually can only penetrate about 100 microns, he's having that penetrate through the entire mouse brain. And he's worked on chemistry in the plastic embedding process, so that the plastic can get all the way through the brain. It's very straightforward stuff, but somebody had to sit down, somebody had to fund him, and some bright guy like Sean had to sit down and year after year do the experiments to get it to actually work in practice. And that's what he's doing, and he's continuing to do that for the mouse. And once he gets it done for the mouse, I think the success that he has already demonstrated for the mouse, and I hope he's going to win the prize in like this year. I don't know. I mean, it's the Cryo people and him are both kind of vying, but I hope whether he wins or not, I hope he demonstrates it, and then moves on to the next scale, which would be, in his case, a whole primate brain.
Hmm. Oh, what do you think the cryo people can learn from the chemical preservation side, and what can the actually, I'm talking about sides again, aren't I? But
Yeah.
like, it seems as though at least there seems to be an
Competition is very,
ideology, right,
yeah,
behind.
competition is very healthy in science in general. Okay, so the idea that there's two sides is not a problem until you pick one of the sides before either one has demonstrated anything.
Hmm. Hmm.
So what would uh, to a large extent, the people that are working on the cryo side, uh, they don't have that much to learn from the chemical brain preservation side. What they really have is they have, to a certain extent, a harder problem. They have a harder problem. They have to preserve the structure of the brain.
Hmm.
They have to preserve the structure of the brain.
Hmm.
But they have also set the requirement that they have to preserve it in such a way that they maintain viability. And so one of the things that the cryopreservation people could do, but they're not going to do, at least from what I've heard, one of the things that they could do is they could apply a light fixative before to the brain before they go through the steps of perfusing cryoprotective agent and getting it down to liquid nitrogen temperatures. They could provide a light fixative that would stop a lot of the damage, the structural damage and the molecular damage that's going to be going on, because the proteins are still there, ready to cause metabolic damage. So if they threw in a little bit of chemical fixative, then they might be able to preserve the structure much better. But they're not going to do that because those cross links, they destroy the ability to just rewarm and bring somebody back, and that is heresy from a lab whose main goal is not to preserve brains, but is to create a kidney banking technique, a technique where you can actually take human kidneys and put them in liquid nitrogen temperature for a few years, and then rewarm them when you need a donation.
Okay. Well,
So what's great? I'm sorry. What's great about this is that the Brain Preservation Foundation has identified these two labs that are really doing this research, and we would love to have more labs. We've got the labs that are kind of doing the best research in the world in these two categories. We've got them. But we would love to have more labs that are working on this research. We'd love to have them compete in the prize as well. But even if they didn't want to compete in the prize, who cares? If they're doing the research, that's a success. And there is many different roads to follow. It's not that there's only two, and we got this lab doing this lab doing this one, and their work is obviously the best that can be done in either of these two categories. No, we should have dozens of labs. This should be a national. Obama should have come out with this never happened, but he should have come out with a national brain preservation initiative. Saying before this decade is out, we are going to be able to preserve a human brain in a hospital surgical procedure, so that nobody has to, so that the information that is most precious to us, our unique identity, is not lost forever due to biological death. And if somebody had the courage to come out and say that, if
Well,
people
you
that are
are.
listening to this video
Hmm.
just start demanding, because this is really, it's a life and death issue. If you're an atheist, skeptic, scientist that buys the what science has told us about the human condition, then you don't go in for praying to God to take you up to heaven. You don't go in for the idea that you have an immaterial soul and that your memories are somehow not connected to your physical brain. You go in for the idea that we are evolved biological creatures, and our brain is a machine, and our uniqueness is the software that's in that machine. And so, if you're in that category, and I know that's not the majority of people, but if you're in that category, then you should be demanding that this research be done. Demanding. Because it's equivalent to having a cure for cancer that nobody's working on. Because, why do we need a cure for cancer anyway? It's ludicrous. People should be funding and people should be researching this.
Cool. Definitely. So it seems like we're just not focusing on the right things. But why? I mean, what's the underlying reason? I mean, why is there so much? Is it people are scared that if they sort of follow this or like advocate this sort of research, their peers will distrust them or not respect them as much. There seems to be a lot of politics in science. Is that something that you think should be discussed?
I think it's less that. I think there's an underlying reason why this is not being researched, and the underlying reason is that it's the same with cryonics.
Hmm.
I mean, this is just a broader definition of cryonics, basically, brain preservation. And the reason is that the techniques to bring somebody back, especially mind uploading, that technique is not anywhere close to being available today. Okay, we can see little hints of technology that could be scaled up,
Hmm.
but it is if we went down and said, okay, when is the first upload going to happen, and when is mass scale uploads going to be happening? It's just not going to happen for another 50 or 100 years. And so, talking about that is kind of crazy. Okay, when I'm talking to one of my colleagues, and I say, okay, we should preserve a brain, blah 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 blah. They'll be able to say, oh yeah, that's not that hard. That's not hard to preserve a brain. But when I start talking about the mind uploading, when I start talking about scanning an entire brain, or if I was talking about repairing an entire brain that was cryopreserved or something like that, their eyes glaze over. And it's because it is absolutely the case that that's science fiction. That ain't here. It ain't going to be here anytime soon. It is something that people have a giggle factor about, because it's kind of like us talking about what we're going to do on a Mars colony, or when we settle the moons of Jupiter, or when we go to the nearest star. You don't want to have a lot of conversations like that because it's way outside of our it's not that people don't believe it's going to happen. It's just that they have a blockage to even thinking about that because it sounds so outlandish.
Hmm.
And yet, to understand why brain preservation is a cure to death that is achievable in the next few years if we worked on it, to understand why that's the case, you've got to be willing to speculate on what future technology is going to bring us in about 100 years. You've got to be willing to make that leap and say, okay, yes, we are going to solve all of these problems. We are going to be able to scan a whole human brain. We are going to be able to simulate a whole human brain. You know, we are going to understand entirely how the human brain gives rise to consciousness in the mind. All of that stuff is going to be done in the next 100 years, or 200 years, or 500 years. Who cares what the actual time scale is? Just being able to accept that that's the case, and get it into your brain that that's the case, which requires, you know, really thinking about future technology and embracing the idea that it's going to be here, and more importantly, really accepting that science really what it's telling us, that we're machines, that our brain is a machine, and we're software. It requires them believing that, and people have a blockage to believing that. But I think that they have the same type of blockage that they would have had to von Braun or to Goddard saying, you know, I'm working on these liquid fuel rockets because, God damn it, we're going to land on the moon. Look at the rocket equation. This is how big the rocket has to be. This is how much thrust it has to be. We're going to land on the moon. And when Goddard's out there in a field with a little model rocket, and he's saying, I'm working on this liquid fuel because that'll create the thrust to get to the moon, it's just giggles and nonsense, and that's never going to happen. Never going to happen. But of course it did, because that's the science. So when people actually believe what science is telling you, and they work toward it, things happen. Great, great things happen. And that's all we have to do in neuroscience. We just have to believe what science is telling us about where we are. Once we believe that, we'll see very clearly that brain preservation is here within our grasp, 5 years, and we have a cure to death. We should just be talking about this with each other.
Sure, absolutely. It just seems as there's, like, according to what you're saying, there seems to be a large disconnect between what scientists know and what science is telling them and what they believe.
That's right. And I think what really would help this along is just having some really good debates
Hmm.
on this topic.
Hmm.
You know, I'm willing to debate anybody on this topic. You know, I would love to get in a room with the, yeah, I'm thinking of these great atheist debates that I always listen to on YouTube. You know,
Hmm.
Richard Dawkins and Sam Harris and
Yeah,
Daniel Dennett.
Deepak Chopra.
Yeah, when they're debating these people, and of
Yeah.
course they probably wouldn't. I don't know how they would come down on mind uploading or brain preservation or any of these things. I know that Carl Sagan, the great
Well, I
Carl Sagan.
do know. I actually I spoke to Sam Harris at the Atheist Convention in Melbourne, and I had a singularity table, and that was a big convention. I mean, like four or 5,000 people or more. I don't know, right? But he was there. He was one of the keynotes, of course. And I got into a small discussion with him about the singularity, about Aubrey de Grey's work, because Aubrey was due to speak at a conference a couple of months after. And yeah, so he actually brought the topic up during his talk at the Atheist Convention, and he said he kind of made a joke about it, saying it was very religious to, like he said, it was a lot of the singularitarians or the futurists are religious in nature, the way that they think. And he said like he'd like to have all this wonderful technology do what some of the futurists are claiming it to do, but he said he doesn't expect his mind to be uploaded into an iPad 25 anytime soon, right? He was kind
Yeah.
of making a little bit light of it. On the other hand, Richard Dawkins spoke about eugenics, and that was very interesting. And he said, "Well, we need to." He said something along the lines of like, "We need to really consider this. You know, we'll be taking these. You know, I guess." He was saying there would be implications to change all of somatic gene therapy compared to germline gene therapy or something like that. And in another interview, he did talk about the singularity and sort of countered his interviewer. His interviewer said the singularity idea is silly, and that AI will never achieve true human style thinking, or will never reach that level. But Richard Dawkins said, well, yes, I don't believe it's impossible. So there does seem to be some differences of opinion in the mainstream popular atheist or skeptic crowd.
Yeah, yeah, and I think and there's other examples of people that really have come to accept. Okay, yes, these things are going to happen in the future. These things are going to happen in the future. If you ask somebody about intelligent life in the universe, they will say yes, there's probably intelligent life in the universe. They'll argue, and then they ask them, well, what kind of life do you think that it'll be if a spaceship ever arrives? And they'll say, oh, it'll probably be machines, because after all, any advanced civilization is going to get rid of their biological bodies, and so yeah, I think that there's when somebody like Sam Harris is saying, oh, I think this singularity stuff is a little bit of nonsense, a little bit of woo or whatever. I would agree with him. I would agree with him. I mean, if you ask me, well, what do you think about the singularity? I would say the timeline is too aggressive. I don't think it's going to happen. Okay, I don't think Aubrey de Grey's stuff is going to happen anytime soon enough for me to live. I don't think artificial intelligence is going to happen that explodes in 20 years or so. It would be great if it could happen, but I think that the timeline is too aggressive. And probably all of these skeptics, including Sam Harris, is saying that. But the question to ask them after that is brain preservation. Should we, given that you said that the timeline is too aggressive, given that you said that you don't think it's going to happen in your lifetime, but it's going to happen. It's not going to happen in your lifetime, but your grandkids are going to see it. To say, to then turn around and say, well, if you preserved your brain using current techniques, just a little bit beyond current techniques, if we put enough into the current techniques, we could get your brain preserved so that all of the stuff that science currently says needs to be preserved would be preserved, and to get there. Wouldn't that be a great thing? And I think, I don't know what they would respond. I think many of them would say, yeah, you're right, but I wouldn't want to. Yeah, you're right, but I'm not philosophically worthy. I can get into this frame of mind, okay? This idea of well, what's the point of little old me getting into the future? I can definitely get into that frame of mind. But if you just ask them, okay, well, what about the possibility, and should people that want that, for whatever reason, to bridge the gap for over 100 or 200 years from now and get to that future, should they have the possibility of getting that? Is it possible that the technology could be developed? I think the serious skeptics, like those that we've mentioned, like Sam Harris, Richard Dawkins, Daniel Dennett, they have, all three of them, talked about really coming to grips with what science says about the brain. We're evolved creatures and the brain is a machine. They've said that so often. They really believe it. They really understand it. They've really internalized that. I think if you laid brain preservation down in front of them, that they would, that they might not personally go for it. I can definitely understand that. But I don't see that they would be skeptical of the very idea. And one of the reasons why I say that is because, and I was going to say, Carl Sagan, in one of his books, Broca's Brain, has this great statement that just absolutely says chemical brain preservation. It's very clear. I don't have the quote in front of me, but anybody that Googles it, you'll see Carl Sagan lays out chemical brain preservation in its entirety right there in his Broca's Brain book. And he was a skeptic. He didn't go in for cryonics. He didn't go in for any of this stuff. But would he have said, I don't know what his opinions of cryonics were, but I'm pretty sure he would have said, gee, I don't think it would work with today's technology. We should probably put more research into it. That must have been his skeptical take on it. And I think any of these skeptics, I'd like to consider myself a skeptic. I know that most people might look at me and say, okay, well, this guy's got to be fringe. He's got to be, these skeptics should do a special on him because he's talking about mind uploading and brain preservation and cryonics and stuff. But they are the natural first adopters of this because they're the ones that have really thrown away the baggage of the religious nonsense of us being a soul, an immortal soul. They've thrown that away. They've embraced the idea that we're evolved beings, and so they're the natural ones that can look at this and look at it with a skeptical eye and an open mind. And I would love to see them address this. And if they're skeptical of it, more power to them. Michael Shermer, another great skeptic.
I've interviewed him.
He is I'm sorry, you've interviewed him, or?
Mm-hmm. Yeah.
Okay. I mean, he was very hard on cryonics, very, very hard on cryonics, but he is a really good open-minded skeptic. So good and so open-minded and so true to his principles, the principles of just test the damn thing and let the data speak for itself, that he was very happy to be on the Brain Preservation Foundation's advisory board. And I'm very glad to have him because he's there to call me up and tell me if I need to be reined in. He's here to keep that skeptical eye. If I say that something works, he's gonna be on the team to make sure that I'm not reading the data with rose-colored glasses. Okay, because I don't want to say that something works just because I want it to work. We want a technique, a brain preservation technique that works because it really works.
Yeah.
And so here is somebody that was one of the most vocal skeptics of cryonics for good reason, because he hasn't seen evidence that it doesn't destroy brain tissue. He's open-minded enough to look at the evidence, and the challenge is out there.
Yeah, so that's great. I think it's good that you have him on the board. Has he admitted up any errors? Has he updated his beliefs on new evidence that you've presented to him that you know of?
The evidence that I have, unfortunately, does not require him to update his belief. The images that I've seen of cryopreserved brain tissue do not meet the standards of preserving the structure of the brain. There is some debate over, and this is to get into details, but I think this is probably a very important detail that your listeners might want to know. So the cryopreserved animal brains that I have seen electron micrograph images from show a distortion of neural structure from severe dehydration. And it is a structural deformation that, at least to my eyes, looks like there must be damage to the connectivity. The researchers that are doing it are also saying, though, that similar distortion, similar dehydration and shrinkage has been shown to be reversible under different cases. That it reverses during the rewarming and rehydration process. Basically, if you take the cryoprotective agent out, wash it out, that these these processes that are shrunk and distorted, they pop back right where they're supposed to be. And I have been going back and forth with them about where is the evidence of this? Is this evidence something that we can test? Is this really the same protocol? And so these are we're really getting down into the nitty gritty of saying, here's what a cryopreserved brain looks like. It doesn't look like it's just a pristine, what you would expect a brain to look like. So prove to me that this isn't a destroyed brain. Prove it. And they've got to prove it because that's what the prize says. They've got to prove it.
So it sounds like to me, actually, I might just quickly. I did actually read some of Carl Sagan's stuff, and he did say something about AI, which was very profound. And he said that people often feel that we have sort of this island of intellectual uniqueness that will never, will never be reached, and computers will never actually get there. But he seemed to be very in favour of the idea that computers are getting faster and faster, more capable, cheaper and cheaper, and smaller, labyrinths repeat. So this is happening, and he said that sometime in the future it's got to be, if we're still around to achieve it, some form of equivalence in machine intelligence to human intelligence. And so it's not like, the human intelligence isn't a magical thing. I can send you the quote later if you like. So I thought that was really interesting. In terms of what Michael Shermer said in my interview, he said that, look, he is in favour of following these ideas through. He's in favour of the idea of the singularity. I didn't talk to him about brain preservation because I didn't know to. I didn't even know he was on the board of the Brain Preservation Foundation. But he seemed very interested in keeping an ear to the ground about these sorts of things. And I think that's healthy because what you were saying before about what it seems to be that we're getting closer and closer to being able to make a connectome copy of the brain that's so detailed that we might be able to revive it in some way, or at least we're getting to that stage. Is that right? And if we do, it'll be kind of like a Sputnik moment, and we might, a lot of eyebrows might be raised, and therefore, it'll be put back onto a lot of the skeptics, and they'll
That's
have
right.
to consider that as new evidence instead
And
of
I
saying,
think if I can put my skeptical hat on for a
mm,
moment,
yeah,
I think all the skeptics would point out that the problem with cryonics is that people are selling a product to people that really want that product because it's they're selling it as a cure to death, certainly.
mm-hmm,
And there is not sufficient evidence that it does what it's not sufficient evidence that it does what it says it does.
mm,
Okay, which is, they're not saying that it absolutely is going to work, but they're saying that it preserves the brain. They're making claims that it preserves the brain to a certain extent, and that the fact that they're selling this product really should raise the skeptical level in all of us,
mm,
especially their customers, which I happen to be one. Okay, and although I may not be, depending upon how these results go, if the results, I'll not become, I'll cease to be a member. If anybody starts to sell chemical brain preservation, and they could, God help them, because they better have proof that it works. And so what the skeptics really should be saying is, don't go out there and sell snake oil to people. There's a possibility that the singularity is basically a bunch of snake oil to make people feel good about the future, and you should be investing in AI because in just 20 years, the AI is going to take over for all of us. Who knows? So to the extent that we talk about these things and we remain skeptical about when they're going to happen and look for milestones and look for actual proof, that is, it should be at least, straight down the line with a good skeptical attitude toward these things. When it starts to veer into, let's say, the cryonics industry, where they say, oh, well, nanotechnology will be able to repair any of the problems, don't worry about it, that becomes very problematic. And so I really think that what I tried to do, I'm not saying that this is perfect, but what I tried to do is I laid out this prize with a very particular set of requirements for winning the prize. And it's the requirements that I think should be able to be met for any surgical technique that could be offered to terminal human patients. If you don't meet these requirements, then you're really asking for trouble offering it to humans. I'm not saying that Alcor should shut down or anything like that. I'm not saying that these cryonics organizations should shut down or anything, but they should at the very least be competing in the prize. They should have called me up day one and said, come on, we've been selling this stuff for 10 years, 20 years. We can meet this like that. We've got reams of electron micrograph data to send you right now. Just tell it, we'll send you all of that, and then you can write us the check for the prize money. And the fact that they haven't done that, and that they have made excuses, the Alcor has made excuses for, we just don't have enough money to actually compete in the prize, that's problematic. That's something that the good skeptical mind should say, what's happening? Why are they not able to show better evidence? So you get the point.
Yeah, but I would say that, if there is an avenue, if people are dying now, and there is no way that to preserve the brain other than alcohol, or other than what a Neural Archives Foundation or whatever institution out there, what is it, the Cryonics Association, whatever it is, CI Institute. So, I mean, people, if people believe that there's a good possibility, that it will result in there being enough information there to, I guess, recreate another a faithful enough version that they're happy to go with, then yeah, they should do that, even though
People
the
should
science is
have the
not there
right.
yet. Hmm.
People should have the right. Alcor should have the right to exist, and the people that are members of Alcor, like myself,
Yeah,
should
exactly.
have the right to decide that they are willing to undergo this definitely untested procedure.
That's right. I mean, you have to take a leap of faith when the science isn't there now, right? If you
Yes,
think the rewards are good enough.
yes, but the skeptics also have the right to
Hmm.
put this in the category of of things that also don't have enough evidence, and also people that may be completely fooling themselves, just completely fooling themselves. And I put myself into that category. So an organization like Alcor should be hyper sensitive to getting the proof that of how well those brains are preserved.
Hmm.
I mean, they should know better than anybody exactly what level of damage they are currently achieving with their current practices. And unfortunately, I have not seen that level of understanding. There's a certain amount of crossing their fingers. Okay, they're doing their best that they can. They are strapped with a lot of undue skepticism and a lot of legal hassling from outside, and very little research funding. And all that I'm basically saying is, my God, we need to get more real scientists involved in this game.
Mm.
There needs to be dozens of research labs with tens of millions of dollars devoted to perfecting cryopreservation. Because do we really want to have this very, very, very important medical breakthrough be getting pennies every year? No, we don't want that. Why do we have this?
Mm.
And to a certain extent, and I've written about this in other places, to a certain extent, the reason why cryonics, now chemical brain preservation is a totally different thing, but the reason why cryonics has not gotten mainstream scientific research is, to a certain extent, blowback from what I've seen, blowback from organizations that were willing to apply this technique to human beings too soon. Okay? Should people have had the right to do it? Yes, I think so. Should scientists have distanced themselves? So there's the Cryobiology Society has bylaws that says anybody that researches cryonics is immediately kicked off. They're not able to publish in their journals. That's nonsense. That's the most unscientific thing I've ever heard of. They should be, looked at badly by the entire scientific community for having said something so unscientific.
Did they actually say that?
They actually said that. Yes, that is
That's incredible.
It is a. You can look up something called from a guy named Mike Darwin, which is fantastic researcher in cryobiology that happens to be a cryonicist, and so has gotten much less than what he is due from all the scientific work that he's done. But he wrote something on this particular thing. It's called Cold War, the fight between cryonicists and cryobiologists. It's Cold War, and it is a great article telling the history from his perspective, and he was there in the 1960s when all of this started happening. How the early cryonics movement was working hand in hand with cryobiologists. They were both enthusiastic about this, and then there was negative publicity from cryonic application to human beings, and the scientific community began to just completely distance itself from it, which is horrific. There's no excuse for that. And so if that hadn't have happened, if the scientific community, the cryobiology community, had not been so vehemently against the application of biostasis for eventual coming back in the future, if they hadn't been so against that for irrational reasons, then we might have had decades of high quality research into cryonics. And if that was the case, we probably would have a cryonics technique that actually brings somebody back to life at this point.
Mm-hmm.
But there's no
so
research
sad.
around it.
Yeah.
So the brain preservation, the original idea that I had to form the Brain Preservation Foundation and to come up with this prize was to break this deadlock. We got a bunch of cryonicists that are working on pennies, trying to do research that takes tens of millions of dollars, hundreds of millions of dollars. This is a really hard problem that they're trying to crack, and they're working on pennies. And we got a bunch of scientists that won't touch it with a 10-foot pole. And some skeptics in the middle that are roaring to say, no, you shouldn't touch it with a 10-foot pole. And what I tried to do is I tried to say, look, if we can come up with a set of milestones, with a prize milestone, where the most skeptic among the skeptics could say, yeah, that's what we want to see. You show us that, and we'll start to take you seriously. And the cryonicists look at those same milestones and say, yeah, we can do that. maybe we haven't done it yet, but we can do that. Then they can start talking to each other. And it hasn't happened yet. It hasn't happened yet. The cryobiology community hasn't embraced cryonics yet, but I think that they should. They should look at those milestones, and they should say, okay, this is the milestone. Is it really impossible to achieve this milestone? The cryobiologist couldn't possibly say, yes, this is impossible to preserve a whole brain. Um, they couldn't possibly say that. And so the Brain Preservation Foundation was really formed to try to break that bottleneck and get mainstream science to embrace this research that so desperately needs to get done.
Hmm. Right. So yeah, it's funny. I've spoken to AI people who seem vehemently against any research into like artificial general intelligence or anything any singularitarian talking about possible futures because they claim it's an embarrassment to the field at large and as such believe that if they associate themselves with this embarrassing factor will decrease the amount of research money that they get and so thereby siphoning the whole pool of research money from that particular industry because of experiments they claim to be wildly imaginative and unrealistic has been like what I said, like embarrassment factors that and I mean that seems to be a common thread in like this cryonics and cryobiology field as well.
Yeah, yeah, it's absolutely true. If uh, people that embrace this pay the price, and maybe that occurs with every breakthrough. You know, there's only one way around it, and that's to just work through it. The scientific method works. The scientific method works. Peer review works. Uh, Cutthroat competition between scientists. I hate it. I hate it. I hate going into to an environment sometimes. When I was in my postdoc, it was a very cutthroat environment where it was definitely I'm going to publish this paper, and the only way that I can publish this paper if this other person doesn't publish this paper. And that theory has to be bad because it's directly competing with my funding that I'm getting.
Mm,
Um, uh, in that type of an environment, uh, being forward thinking and embracing these types of potentialities can be absolutely career ending. But what are you going to do? People are people. Uh,
mm,
all you can do is just continue to put forward the best argument that you can and work on the things that you think that you can work on.
mm,
And I know there's a bunch of closeted transhumanist mind uploaders in the neuroscience. I talk to
mm,
them all the time. You know, they believe all this stuff. They're enthusiastic about this stuff, but damned if they're going to start working this into their conversations with their colleagues.
mm,
It'll be death for their career,
mm.
or at least they believe it'll be death for their career. So uh, I have no easy answers.
There are none. But this seems to have happened. I hear words that this seems to have happened in other industries. I mean, the first person to really talk about dark matter, or maybe not the first person, but one of them, got ridiculed a lot. Um,
Yeah.
or dark
Yeah.
energy, and yeah, so there's been prevailing scientific opinion that in order to continue funding in a certain area, there needs to be a mantra that's sort of like repeated over and over again, and that's what funders like because they know that this mantra has proven true for so long and has seemed to have caused so much funding for so long. Why should we stop that? But maybe it's about education. I'd be surprised if we don't get better at this, or we don't get better at spotting this sort of issue in the future. I mean, a lot of the research now is a lot more audacious than what it used to be, right?
Yeah, and there's it's interesting. There are several examples. Like for instance, I mean the idea of cosmology. Um, you know, I've heard lectures, they're quite common now, where people talk about how in 100 billion years, I think it is, we won't be able to see other galaxies because the dark energy will have pushed the expanding universe out. And these are these concepts are so outlandish, are so futuristic that they make mind uploading seem quotidian. They're just uh, and there's no problem with talking about these.
Hmm.
So actually, I think that it will just switch at some point. At some point, it'll be obvious that brain preservation is a good thing to work on. It's just obvious, like a cure for cancer. Of course, you're
Hmm.
going to work on a cure for cancer. Of course, you're going to get lots of stories that are excited about this possible cure, this possible cure, and this possible cure, and some of them might be very out there. But
Hmm.
if you had a cure for cancer, it would save so many lives that and so much suffering that it's worth trying
Hmm.
as long as the theory makes somewhat sense and it makes sense to devote some research dollars in that direction. Uh, People will eventually see brain preservation this way. They'll say, look, this is worth trying, and mind uploading or repairing cryo damage and things like that. It's outlandish right now, but it might occur, and this is so important that we should definitely just work on it.
Hmm.
I don't know what it's going to take to do that. I
Well, the
think what it's
Russians
going
put
to take.
Sputnik into space, and then the Americans got like really scared.
That's
Um,
right. Although there's not going to be a Sputnik moment, as Obama says, for this, because basically the Sputnik moment will be 50 or 100 years from now when it actually works. It's not going to be obvious that you have a brain preservation technique
hmm.
that will has obviously saved the person. You're going to have to trust your science. Just like now, extinct species or species that are going extinct, they'll save some of the DNA.
Hmm.
They don't know what they're going to do with the DNA. They haven't proved that they can bring back a woolly mammoth or something like that. They haven't proved that yet. But it makes so much sense. They can see the writing on the wall that they say, yes, we should definitely start preserving this stuff. Because down the line, people are going to get really pissed off at us if we didn't go to the little bit of trouble to save some of the DNA of these extinct species.
Hmm.
Yeah.
Hmm.
Um, and so this is brain preservation will definitely look that way at some point. It may take quite a while. And one thing that it's going to take is it's going to take people with the courage to put their careers on the line. And I don't have that much of a career. Maybe I could have. I don't know, but I don't have that much of a career.
Yeah,
So
right.
it's if I was a big name scientist with a big lab and a lot of funding, and I believed in this stuff, I would probably maybe I would keep my mouth shut, but I'd like to think I wouldn't keep my mouth shut. And if there's some scientists out there that actually do have the clout to put their reputation behind it, that will significantly move the ball forward.
Yeah. I wonder if in the future, like, when and if brain preservation or something like it is justified and vindicated, all these big scientific institutions will turn around and say, "Hey, look, we've been saying this all along, right?"
Absolutely, absolutely. It happens all the time in science. You know?
Oh.
I remember, Isaac Newton. Isaac
Mm-hmm.
Newton got made fun of by one of his colleagues or something for having the gall. He's like, oh yeah, this Newton guy. He's great about calculus and stuff, but he's starting to talk about this about how the moon creates tides, and that's utter nonsense. And he obviously put it in the category uh, of of werewolves. you know? You know, the moon having
Yeah.
effect on tides,
Mm.
but now it's just obvious. It's like, okay, of course, the moon has tides. It creates tides. It's in textbooks. Yeah.
Mm.
So now for some crazy reason, it seems outlandish to talk about preserving the structure of the brain so that we can one day image the whole brain and put it in a computer. It sounds outlandish. Sounds
Mm.
totally freaking outlandish. But uh, we have to drill into why it sounds outlandish. You
Mm-hmm.
know, if somebody could send me point by point why it's outlandish, and people send things all the time, but they don't know neuroscience. They're not really, they're not making the proper connections with the real stuff. But if somebody is out there that really sees the fundamental flaw in this whole thing,
Mm.
and you send it, I'm open-minded. I would like to be put right. I can toss this aside. I was wrong about the whole thing and move on with my life.
Mm.
That's what any scientist has to do. They have to take
But
what
do
they're doing,
they?
and they have to be willing to junk the whole damn thing
Mm.
when evidence appears that says it's wrong.
Well, on that subject then, first of all, has anybody sent you any information which what has been the closest to convincing argument against preservation that you've encountered?
Well, it's a little hard to say because it's one has to start with the idea that we don't have a preservation technique that works. So it's really it wouldn't be a convincing argument that preservation doesn't work. It would be a convincing argument that achieving preservation would be extremely difficult. Uh, and so at the beginning, when I was considering chemical brain preservation, the idea of chemical brain preservation, there were several scientists that really took issue that there would ever be a way to get a large volume chemically preserved for electron microscopy. Okay. And they pointed out that osmium just can't get in. I remember one that said, "Look, I've tried this, and osmium just can't go through a large piece of tissue. It just won't work." And I pointed to some papers, but that might be evidence, but this was a real scientist that had done these experiments, and I took her very seriously until good old Sean McCoolah totally showed her wrong. because here is a whole mouse brain that is preserved with osmium fixing lipids, osmium fixing its lipids all the way throughout. And so yeah, there have been some there's been some skeptical voices that I've taken very seriously, and I still take them very seriously, but none have offered anything like this is so difficult as to be not worthy of pursuing in earnest to get it done over the next few years.
Well, have there been any other sort of beliefs that you once held close and dear to your heart that you had to overturn and discard in light of new evidence?
Uh, yes, Yes, I'm trying to, yes, tons. I am definitely, uh, I am not somebody that clings on to beliefs. When I, so I'll give you a for instance. I'll give you a for instance. So um, maybe this might not be the best for instance, but when I went into graduate school, I was uh, in neuroscience. I got the chance to work with one of my idols, a guy named Irving Biederman, who had developed a theory of how we recognize objects, and and I really loved that theory, and I really wanted it to be true, and I designed a whole bunch of experiments to kind of test its internal workings. And I'm not saying that I've dropped down, proven it wrong or anything. I'm sure that he would disagree, or but there is some aspect of there was a particular aspect of it that I really wanted to show was right. And every experiment that I did just showed the opposite. Yeah. And so I had to basically come up with a PhD thesis that went in a completely different direction than what I started, and that's fine. It's progress. I mean, what hubris would I have to think that I would have known uh, uh, how this was supposed to come out when I got into graduate school? And so I think I have I've had some shaken beliefs with how fast nanotechnology is going to come about. I was very enthusiastic about nanotechnology when I read Engines of Creation by Eric Drexler. I read his Nanosystems book cover to cover. I was very and I still think nanotechnology is going to be here, but the time scales I have come to believe are very difficult. There's tons of these. I once was Christian. There you go. I was once
Okay.
Christian,
Well,
and and
a lot of the people I've interviewed have been and are.
yeah, I mean, I would debate. I gave it up in high school,
Ah.
I guess, but I very much believe I was an altar boy at some point,
Is
and
that why
so
you
there's
left?
that completely got demolished. I have a bunch of political beliefs that completely got demolished. I, hopefully this is not going to be one, but who knows? And it could be, the Brain Preservation Prize. I hate to say this, but it could go unwon year after year after year after year after year. If somebody doesn't meet the requirements of the prize, it ain't gonna be won. So it is not a prize that's just to put a stamp on something that I like. It has to get through a round of judges, and it's got to be published, and these are the requirements for the prize. It's a very skeptical-minded prize, and if I believe that it worked, that ain't good enough. It has to go through some serious rigor to work
Hmm.
to pass the muster to win that prize.
Hmm. Hmm. Okay, so you mentioned Aubrey de Grey before, but it sounds like uploading would be a cure for all in a sense. Do you see this as happening much later than the biomedical realization of Aubrey de Grey's SENS research approach to rejuvenation through repair of accumulated damage?
Yeah, so so I'm opinionated on this, but my opinions are probably, they should be taken with a big grain of salt because, Aubrey de Grey is an expert in the field of biochemistry and aging. I am by no means an expert in this. As I speculate on these things, though, what I see is I see very difficult problems to curing aging. And so, uh, if I had to, if I had to guess where a significant life extension is going to occur. When are we going to get the technology where we can really fundamentally extend human life by 20 years, let's say, yeah, or by 30 years or 40 years or something like that, something significant, something that's just not just, a little bit curing these diseases, but to really extend the quality of human life. I have to say I put that out many decades. I don't see it happening anytime soon. It'll definitely happen, but I put it out many decades. The mind uploading stuff, I put that out at least 50 years, at least 50 years, and that's for the first demonstration.
Yeah, right.
That's not for the run-of-the-mill person. It's out there. The nanotechnological repair of cryopreserved patients, my God, if it's possible, then that because of the good cryopreservation, where do I put molecular nanotechnology out there? These are such difficult problems that I would have to put it out many, many decades. The demonstration of reversible cryopreservation, this is something that the Cryonics considers as the holy grail, and it's something that the skeptics have put out as, show us this and we'll believe you. That's easy to say, okay? When are we going to be able to take a dog, bring it down to liquid nitrogen temperatures so that it could stay there forever, and then rewarm it and have the dog walk off the table and live a normal dog life? My God, that is a hard problem. It is a super, super, super hard problem. We're talking decades. When I'm talking to the people that are the most enthusiastic about this, they say decades. Okay, so all of these technologies, and general artificial intelligence, man, we should be farther along than this. But I don't know. That might take decades. I would actually say that should come a little bit sooner. But all of these things are well beyond what I consider to be my possible lifetime. I'm going to miss every single damn one of them. Okay? The only thing that I'm going to see in my lifetime is normal human beings are going to, private citizens that are not NASA astronauts are going to walk on the moon, and they're going to walk on Mars, and they go to space stations. They're all going to be rich. And I'm going to miss that too. So all of this stuff I'm going to miss. And which sucks. And I should be jealous of my kids. Okay, because they're not going to miss any of this stuff, hopefully. But when I look at this portfolio, I basically say, if there is any hope, it comes from cryo or chemical brain preservation. And so if I had $1 trillion in my pocket, guess which technology I would spend it on? Okay, I would spend it on I'd spend it on the horse that is most likely to cross the finish line before I'm going to die. And so that's why brain preservation is such a I mean, I've talked to some people, that say, oh, well, look, why should we care about brain preservation? Because, let's say they're in their 30s and they've been convinced that the singularity is going to occur long before they need cryonics or anything else. So why should they care? I think that's I think that they're being overly optimistic. And by the time that they come to the realization that this technology was farther out than they thought, it'll be too late to
invest the money
to invest in the scientific research that could have gotten them that extra time.
Well, I think it's interesting to talk about then some accelerators and decelerators to these technologies. Specifically, I mean, sure, it's easy to say funding at this stage, and I think we all know that funding would be an accelerator. But Can you go into more detail specifically on exactly what you have? You've spoken about particular research subjects, so maybe it's a moot question. I don't know. Is there
Well,
anything else that you'd like to bring up though?
yeah, sure. On the cryo side, there is it's definitely the case that the current cryopreservation techniques uh, are are they are creating a type of distortion to the tissue that makes it difficult to evaluate damage or no damage to the neural circuitry. So research needs to go into verifying the what the level of damage is for current cryopreservation techniques. Okay, that is a clear research goal that is underfunded right now. There's plenty of funding for, can we, there's not plenty of funding for any of the cryobiology stuff, but there is funding going into demonstration of ice crystal formation, things like this, but there isn't as nearly as much as that's going into actually looking at the circuitry changes, the structural circuitry changes in the brain that are being caused by the best cryopreservation techniques. Research needs to go into that. On the chemical preservation side, it is really all about getting the chemicals that we know preserve structure of the brain, getting them to the right places, getting them to the entire brain. So if we have a cubic millimeter of brain tissue, if I've got a little cubic millimeter of cut out cortex tissue, we've known for 40 years how to preserve that in all of its glory in plastic forever. We've known how to do this. If you're talking about something that's the size of a mouse brain, McCoolah has a protocol that is very close to being able to just demonstrate that that can be done. Okay, getting to something larger than that, even a rat brain, you can't use the same technique. It doesn't look like, at least, that you can use the same technique because it's using simple immersion. The way around this is to use the vascular system of the brain to get a lipid fixator into all the cells, to get around to the diffusion barrier, and also probably on the plastic infiltration steps. So these are very concrete research topics that somebody needs to spend a few years of graduate time on, really doing the hard work and the hard experiments, lots and lots of failures, and lots of ideas tested to figure out what the protocol is. So both of these things, these are concrete things. Somebody, this is not, we don't have any idea what we'd even fund to get this done. This is here is a research proposal that is just waiting for somebody to do.
And it's surprising nobody's doing it then.
And it's surprising nobody's doing it. Yeah. Or the labs that I'm talking to, they are doing it, but they are they have a bunch of other stuff that they are doing as well. And
so they don't have enough time to spend doing these particular experiments.
Well, let's put it this way. Let's take McCoolah. He's very interested in. He wants to win the Brain Preservation Prize, that's for sure. And to do that, you have to show a large mammal brain. But he's in a laboratory that is dedicated to getting a whole mouse brain connectome. That's their long-term goal. It would be a fantastic goal for neuroscience if they could do that. Mm-hmm. He's probably got the protocol that is needed to do that. Are they going to turn around and say, hey, let's fund you for a couple of years just trying to apply this to a human brain? No, they're not going to do that. They're in a lab that's working on mouse brain neuroanatomy. They're not gonna scale this up to a human being.
Mm.
And so some other thing, some other vehicle has to take this, maybe through him, maybe through somebody else, but has to take this to that level.
So, if you let's just say somebody does reach this goal and achieves the conditions to win the Brain Preservation Prize, Foundation Prize, do you have any vision as to what could possibly, if you had the funding, what could possibly be the next prize?
That's a great question. That is a great question. So what John Smart would say is uh, he would like to see a set of prizes that actually uh, kind of pushes hospitals, or not even hospitals, it pushes the implementation of this. That actually gets, it's one thing. So the Brain Preservation Prize is set up so that the final stage of the prize is demonstrating on a large mammal, like a pig, that this procedure works in a procedure that could be applied to a human in a hospital setting. That's the requirements of the prize. So the idea behind this is that a researcher that there's no problem with doing this to pigs. Nobody's going to be say, oh my God, they're not treating their pigs correct. Well, maybe they would say this, but the idea that we're preserving pig brains is not going to cause eyebrows to raise. Okay, nobody's advocating. The prize does not require this to be demonstrated on a human. So there has to be, after that prize is run, somebody has to take this technique and actually implement it in a surgical procedure that would work on human beings. Now, what I would like to see happen at that point, and we're speculating because this may never happen, and maybe it's impossible. Maybe I just don't know that it's so difficult. But what I would like to see is that prize is won. The milestone is absolutely proven that it can preserve the connectivity of the brain. And then medical doctors start to really turn this into a procedure that can be applied in a regulated hospital setting. Okay, none of this backroom abortion crap that that Alcor basically has forced upon it. Okay, we want this available in the regulated, licensed medical hospital settings, just like any other surgical procedure. That's the real goal. And so, would that be a prize? I'm not sure if that works itself into a prize. I'm not really a prize type of guy, believe it or not. So the other thing that you could imagine, though, is that there are people that argue that the prize goal of structural preservation may not be sufficient. I'm sympathetic to this. I don't think that that's the case. I think that it is a good milestone based upon everything that we know about neuroscience right now. That if we had the molecular structure, the structure of the brain preserved, then that would be fine. But it would be great to also have the molecular structure preserved as well. And at least most of this, from what I've seen, is preserved in this chemical preservation technique, and certainly in the cryopreservation technique, that you've got ion channels and receptor proteins and things like that are preserved, but some of them are probably lost. There could be another prize for doing better brain preservation. Get me something that gets better and better and better and better every year. Because we shouldn't just be satisfied with the bare minimum. We should want a technique that is more reliable, more stuff is preserved. It should get better and better and better until it's so good that you can bring somebody back.
Excellent. So, were you involved in the whole brain emulation roadmap?
Yes, I was. Yes.
Yes, right. Were you satisfied with the roadmap?
Uh, no.
What's missing?
I was a vocal
Did
member. of it.
Yeah.
Of course, I
Okay.
was satisfied that
Oh
I
yeah,
loved
it's a
what
good achievement.
Anders did with it. And, um, my main qualm about it was that it was too vague in terms of uh, the the level that needed to be emulated.
Hmm.
So the roadmap basically said, well, it could be that all you have to get is the structural connectivity of neurons, or it could be that you can even have less than that, or it could be that you have to get every single molecule in place, or it could be that you have to get the quantum mechanical structure. And then they basically said, yeah, we probably think it's somewhere in here. Uh, I think that that we that neuroscience and cognitive science are advanced enough fields to not be so questionable about that. I think that we have a much better understanding of how to bracket what needs to be emulated to
Mm-hmm.
get a person,
Right.
and that was my main problem with the roadmap.
Right. Okay. So do you think it should be revised? Would it be a worthy endeavor to make a 2.0 version of the roadmap?
Yeah, I think it would be. I think yeah, yeah, absolutely.
Right. What would a more detailed version of the whole brain emulation roadmap do generally? What would it achieve other than just, satisfying the people who made the roadmap a little more? What would be the flow and effects?
Yeah, well, I mean, if I can just talk to the connectomics side of this,
Mm,
okay. So I helped write the part of the roadmap that was dealing with connectomics, with tracing neural circuitry and stuff. And the the techniques have changed so much since that I think at the very least that we've come so far along. I mean the progress that has been made in terms of uh, so let me give you for instance. I mean, so I work at Janelia Farm, Howard Hughes Medical Institute, and we have been running focused ion beam SEM for the last 3 months straight, getting a large chunk of the fly visual system
mhm,
that has the three main components of the fly visual system, and we're getting this at 8 by 8 nanometer resolution, so that everything can be traced. And Janelia is a great place to work at. They've got they've got the best software people that have created this pipeline that can take that really high resolution SEM data and trace the circuits almost fully automatically, and then have all these other software tools that can allow very fast human correction of the data. And so there's this pipeline that really takes large structures that are large brain structures, like the fly visual system, and goes all the way to tracing every single synaptic connection within it that we now have available. And we didn't have that back when the roadmap was created. And so all of these technologies have really been greatly accelerated over the time since the roadmap was created.
which
So
wasn't all
just
that long ago.
which wasn't that long ago, yes. But Connectomics wasn't. So the invention of Connectomics. If I had to put a date to the invention of modern connectomics, it would be Winfried Dink's 2004 paper on his serial block face SEM. Uh, that's not that long ago. So the whole idea of massively automated uh, three-dimensional electron microscopy of brain tissue has not been around for that long.
Hmm.
And yet there is tremendous progress in it because once Winfried designed that machine, the algorithm people that could analyze the data came out of the woodwork and said, "Oh, we're finally going to get neural circuits. We're
Hmm.
not going to get these Golgi images that are kind of vague. You know, there's a neuron here and a neuron here, and they may talk to each other and they may not. We're going to get the
Hmm.
entire circuit diagram." And that has caused a tremendous excitement in the neuroscience community because they finally get they may finally get the ability to see the stuff that they've been speculating on for decades.
Oh, that's great. So, yeah, well, I mean, like, is it something that just needs funding to happen, like an update to the whole brain emulation roadmap, or um, is it? I mean, I can't see any reason why it would be just completely out of reach. Why wouldn't it happen? Why?
Oh, the specifically the whole brain
Yeah,
emulation.
that's right. Yeah.
Oh, I do not know. I would ask Anders.
Yep, 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 definitely. All right, well, maybe that's probably worth doing, asking Anders. But anyway, I mean, like everybody's short of time, it seems. You know, how can you do everything at once? But how much time would it take, do you think, to update the whole brain emulation roadmap? What's required?
Uh,
How much effort, time?
so actually, I don't know. It all depends on on what you wanted to get out of it. I will say that I've considered writing a book. At some point, I want to write a book that lays out not only the ideas for brain preservation, but for how I think mind uploading will occur. The goes all the way from the theories of the brain to the theories of consciousness, and really lays out the whole thing. And
Hmm.
I've thought through how long it would take me to do that, and it's quite an effort.
No small
And I,
task.
I think that that's really, I mean, I'm obviously I'm talking from my own perspective here because a lot of people don't even agree that whole brain emulation can be done from a structural uh, uh, map of the neurons in the brain. So, you know, what I'm saying is that it would be nice to have a roadmap that not only talks about the technologies that are out there, but really starts with an understanding of what are our theories of consciousness? What is this identity that we're trying to bring back
Hmm.
and to transfer? What is the best theories of how the brain works on that level that can support this theory of consciousness. Okay, there are the production system models of John Anderson Actar. I think that is the best model for this. These are very concrete cognitive science. Dozens of labs working on these on this particular theory. That is definitely something that's totally missing from the roadmap. This idea of what is consciousness and how is it implemented in the algorithms of the brain. This is where you should start, in my opinion. Once you have that, the question is how is that implemented in the brain? So I actually did a first take, a paper on this that I published in the uh, some weird name journal. The I'm blanking on the name of the journal. Anyway,
That's all
there
right.
is a special issue
It wasn't
of
SJN, something like that.
that was devoted to the topic of mind uploading.
Okay.
And I wrote a paper in that tried to briefly touch on all of these issues, all the way from what is consciousness through to how could you extract it from a chemically preserved brain using focused ion beam scanning electron microscopy. I tried to cover that whole breadth so that you really did have kind of a roadmap for a particular technological uh, way to get there.
Mm.
Um, and it is Journal of Machine Consciousness.
Okay.
Journal of Machine Consciousness. Yeah, and it was a special issue on mind uploading.
Awesome. I might look it up. Oh, look, I'd say it'd be awesome to get to kickstart some other like a new initiative to update the whole brain emulation roadmap. So yeah, I mean like ballpark figures. Would it cost 2 million? Would it cost one, 500,000, a billion?
To make a roadmap,
Yeah, to upgrade the roadmap.
I I think it would just take a really dedicated researcher with a lot of time.
Oh, right. So you don't think it would be an issue of getting the right people together? It's just, if there
Oh,
was one person.
yeah, I think it would be one person. I mean, you're just asking my opinion after all.
Yeah,
So,
of course.
I
Right.
mean, so uh, given that open source software can do everything remotely, I would expect that a bunch of internet conversations could gather all of the information that would be necessary to compile this. Maybe that's naive. I have never put together something like this, so.
Okay. Um, well, okay, let's talk about like updates to the definitions of certain things like death, especially
Mm.
over the years. Do you think the definition needs or could use a an upgrade itself? Like, is the definition of death stopping progress in achieving some form of like a suspension technology, like plastination or cryonics?
I think that's a great question. I'm going to go off in a different direction
Hmm.
than I think you expect, though.
All right.
Uh, the definition of death is actually much more problematic than just the idea of when death occurs. Uh, and this is easy to see when you really think about mind uploading and you think about, well, what if you, instead of creating one copy of the original person, what if you create two copies?
Yeah. Hmm.
Then what the heck does that mean? And we've got to just get beyond this, gee, the intuition is the copy isn't me stuff. Because that's an intuition that's just wrong. It's goes against the idea that we're mechanical beings, and of course, mechanical beings can be copied, and there is nothing missing from the copy
Mm.
because there's nothing to miss. Uh, and so once you get beyond that, then you have to accept the idea that if you had a copy right in front of you, uh, you would not necessarily know that that's you.
Hmm.
You might believe that that's not you. If you believe some of the, it depends on how you think about it. But certainly, if that copy of you got poked in the arm, you over here wouldn't feel it. And so, what do we mean by you? Now, philosophers have talked about this in the past, and there is some very good philosophy on this. And I think probably the best philosophy on this is Derek Parfit, and blank on the name, but there's another philosopher who wrote a book called I Am You. I'm blanking on the name of that philosopher, but these two have looked at the idea of our conception of the self and have come to the conclusion that I think I've come to as well, that we really have a screwed up notion of what the self is. It's a screwed up notion that has been built into us by evolution, but it is a screwed up notion. I wrote an article. It's on the web. It's called "Killed by Bad Philosophy." It goes through my take on these philosophical issues. But in essence, in a nutshell, what this says is uh, the whole idea that we are absolutely different from other people is a kind of a naive idea. That is, who knows where it came from? Maybe it came from Christianity. It probably just came from evolution, wiring our brains this way. But the idea that we think that um, that when we die, that death um, uh, is really an ending of something that was completely unique, that is wrong. That is wrong. Uh, what we really are is we are machines that have very unique personalities and memories, but they're not completely unique. And our uniqueness is only that which has been built up over our lifetimes and our genes and stuff. And so, to a certain extent, we're not going to die when we die. And that sounds
Yeah, I
very
know what you mean.
that
Yeah.
sounds very crazy. Okay, I shouldn't even. I'm not going to be able to do this justice.
Mm.
The books that do justice to this are IMU,
Mm-hmm.
uh, and and Derek Parfit's book,
What is
Persons
you?
and Reasons, I think, Reasons and Persons, something like that, and it's a conception that I think is where you're led to if you really believe neuroscience. If you really just take a look at the science and see where it's going,
Mm-hmm.
you end up in a place where our basic conceptions of self are a little bit wrong.
Mm.
Now, does that mean that we shouldn't be afraid of death? No. Does that mean that we shouldn't preserve our brains and try to, or does that mean that we shouldn't, get chemotherapy so that we can live, more decades if we get diagnosed with cancer? No, it doesn't mean any of that. It means that we are preserving our special uniqueness, but it also doesn't mean that we should be looking for this ultimate uniqueness that is beyond even our memories and our the stuff that we've built up that's unique over our lifetime.
Right. Okay. So, I guess in the far future, speculatively, we could sort of connect into like a big sort of network of minds, like a hive, a big collective. Um, and as such, if the network speed between sort of thinking entities increases far beyond what verbal communication does, even beyond the speed of like a far future, like uploaded minds could easily probably connect to each other much easier than like a biological minds could. So, do you think that do you think there's anything interesting there? Do you think people would lose the important, unique aspects of their selves, or do you think that that could be preserved as well as being able to share richness across, yeah, many of the minds in the collective?
Uh, so I think definitely uh, there's definitely some truth to that. I think that we will be going in that direction at some point. The exciting thing is that now we're really talking about something that I have I have no conception of what's going to be possible, what's going to happen in the future. The when we really have uploaded minds and we can do anything we want, that first of all we understand ourselves like we understand our technology today, that we have the same level of engineering understanding of ourselves at all levels, consciousness to molecule, as we do to the computer that we have today. Uh, the the things that we could do are just mind-boggling.
Mm.
And I think that excites me, and the very idea of understanding. See, we shouldn't be surprised. Maybe we are, but we shouldn't be surprised that neuroscience and cognitive science have overturned some of our intuitions about the self and about what's possible and what's even desirable. The fact that we're overturning those intuitions now because of what we've learned in science, and the fact that we're going to turn that into engineering practice
Yeah.
in 100 years is just radically exciting, and it just makes me want to be there. It makes me want to be there. But even if I'm not there, it's still exciting. It's still something that we should be working toward. And it makes me question the idea of, okay, if I'm there, what does that mean that I'm there? What does it mean that I'm there? It does beg a question. Okay, so let's say 20 years from now, I go in for a brain preservation technique. Was that the kin from 2013? No, because that 20 year from now kin has very little to do with me. And so that's yet another level
It's like a continuum.
of understanding of who we are. That we are, yeah, we are a continuously changing process. And so we're dying every day. We're dying every day. Our uniqueness is being erased as part of the normal biological process and being replaced with other uniquenesses, which is great. But I've been playing with my kids recently, and it struck me that that I was very sad that some of the stuff that I used to like and do when I was a kid, I don't like and I don't do anymore as an adult. And I felt a little bit of mourning for my past self. I think everybody can appreciate this. And so, to a certain extent, that's great. That's part of the process. We should love that. Maybe that's one of the reasons why we should embrace death. Okay, I'm not one that embraces death, but at the same time, I can understand people that do. I can understand people that do. But in the future, when we have when we have mind uploading, even that may be gone. If you want to recall exactly your person when you were 19, bam,
There it is. Yeah.
there it is. And so the idea of connecting in a Borg hive, the idea of having yourself called up, the idea of mixing minds, all of these things will be open.
Mm.
It is a fantastic world that we're headed toward.
Mm.
And
Yeah.
in the grand scheme of things, if we don't make it there, if we make it there in 100 years, and nobody adopts brain preservation technology until 50 years from now, and so all those people, all of that uniqueness dies, is lost forever. That's sad, but it's not sad on the grand scale of things. On the grand scale of things, we need to make sure that we, as a species, achieve this. It is mind uploading to me at a basic core is something that the human race deserves better. It deserves better than pain and suffering and aging and dementia. It deserves better than that. And whether we achieve it in 50 years or 100 years or 200 years, that matters less. Or whether a person today lives or gets preserved or doesn't, that matters much less than that the human race gets there eventually.
mm, mm. I've interviewed a few people, and I've had a similar sort of a view or concern or worry. Marcus Hutter, you know who he is, right?
Mm.
Marcus Hutter, he's in Australia. He's in ANU, and he developed the AIXI algorithm, the universal AI sort of mathematical formulation. But anyway, what his less species oriented. He's more the concept of intelligence. If we get to a stage where we can make an intelligence that can continue on and reproduce and do wonderful flourishing things, then that's important. What he hopes won't happen to the universe is that it'll be devoid of intelligence at some stage. Yeah.
Yes,
Mm.
yes, yes, and that is it's uh, yes. So we should fund asteroid detection
Yeah, 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 yes,
more
yeah.
than brain preservation, even.
Well, I mean, to me, it comes down to being able to be resilient under pressure and building resiliency into our society, into like our technology, such that a solar flare won't, take us out and send us back to the Stone Age or wipe us out completely, or that we're not, killed by like a rampant like bacteria swarm that most likely won't evolve to intelligence in the future. So yeah, I mean, it's we have the foundation, we have the sort of the networks now, we have the scientists, we have the society that can develop these forms of technologies now, and we should take advantage of it.
Yeah,
It's really
yeah,
like
yeah. And
like
these are echoes that have gotten a little bit. Uh, so I mean, if we turn back the clock 20 years to the Cold War,
mm,
then this is what all the scientists were saying. They were
mm,
saying, "My God, do not go to a nuclear Armageddon. It's
mm,
just not. That is the thing that must be avoided at all costs." And that sentimentality should still be with us because, believe it or not, there are other dangerous things than human beings.
mm, definitely. Oh, I'd just like to ask a couple of questions about like recent technologies. What are your there's open worm. You've heard of open worm, and
Yes. Mhm.
yeah, yeah. So, do you think it's worth trying to create a whole simulation of the C. elegans? Um,
Yes, absolutely. The one thing I would say
mhm,
is that so when you talk to the people that actually got the connectome of C. elegans,
mm,
they will very clearly say that they do not have much of the stuff that would be needed to be even called a connectome. Um, in from an emulation sense. They don't know what the neurotransmitters are. They don't know the signs of the synapses, the actual functional connectivity uh, of C. elegans. And so that's why this is a difficult problem, even though we have ostensibly the map of all the connections in a C. elegans nervous system. We don't have the functional connectivity of that. If you were to take a piece of mouse cortex and have the same structural understanding of it, you would know also all of the functional connectivity, or at least a lot of it, from electrophysiology that's been done. But it's harder to do that in C. elegans. And so David Dalrymple's work
mhm,
on and others,
yeah,
a lot of others on really uh, understanding with the new techniques with optical imaging techniques and other techniques, understanding the functional connectivity that goes along with that structure is important to make any of these things actually work in practice.
mm, definitely. So it's a good thing to do. Yeah, I interviewed David Dalrymple. He said he's looking for some footage, some video footage, so I can use for B-roll for the interview I did with him. It was quite a good interview, high def and all that sort of thing, because I had my good camera there, so that will be forthcoming. Um, and the other question would be, there has been what's been titled as the world's first complete computer model of an organism. That's every molecular interaction taking place in the life cycle of the Mycoplasma genitalium, and that's the world's smallest free living bacterium. Is that it? They say that that's been completely computerized or not uploaded, but it's a complete, I guess, functional representation of the Mycoplasma genitalium. Is this something that you see as an important development or profound even?
Uh,
Are you aware of it?
oh yes, 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 absolutely. I mean, it's stunning. It's absolutely stunning in its own right. Um, it is marvelous that we have that level of understanding of a whole organism, and the molecular machinery is to a certain extent is more complex than the nervous system.
Hmm.
There's the levels of details of the machinery and the interactions and even the logic circuits that go on in genetics are just incredible.
Hmm.
And so it is a wonderful achievement. The Is it profound? Uh, no, not if you. It's profound in one sense, but it's like of course they could do that. Did they really suspect that you couldn't do that? It's like was landing on the moon profound? Yeah, landing on the moon was profound, but not because we didn't think we could do it. It was profound because we actually did it. And so it's profound in that sense. Um, uh, Now, I think the real question that you might be getting to, though, is does this have anything to do with whole brain emulation? And I would vehemently say
That was the next
no,
question.
because the level at which the brain operates is uh, is not connected to the level at which the machinery in the cells process the DNA and move things around the cell and the cytoskeleton, etc. All of those processes, unless the neuroscience books are just completely wrong, they're behind the scenes to make it work. But to understand it's working, the algorithms that the brain performs, you wouldn't really need to understand those. Basically, if you understand Hodgkin-Huxley equations of neuronal propagation, you're probably already at a level that's too far down when you're talking about the algorithms that the brain performs. Um, so it's great that we have this knowledge, but it's kind of irrelevant to whole brain emulation.
Hmm. Hmm. Would you consider yourself a transhumanist?
Yes.
Okay. Why? What do you identify with transhumanism?
Well, I mean, the main thing that I identify with transhumanism is mind uploading, but I've met a bunch of transhumanists that disavow that. So um,
Well,
uh, I I would I would embrace that title because it seems like that title is associated with a lot of forward, open-minded people that are expecting that there is something better for humanity. That humanity should not be satisfied with what evolution has dealt us. And so
hmm,
that's why I'm a transhumanist.
excellent. And what are your views on the singularity then? Do you think, I mean, obviously, I think you said that you think it's possible, for a machine like a general intelligence. Are you in favor of like the idea of a massive spike going up exponentially for a very long time? Or you think it'll be like more Maximore's model of series of surges? Or do you think it's not possible?
I haven't given a lot of thought to this.
Mm,
Um, I really have not given a lot of thought to this. I think probably the reason for that is that I kind of think that these things will uh, uh, that will muddle through each one of these. That it won't be an overnight type of a radical change in the world.
mm,
Um, we look back over the last 10 years, and the internet has taken over all of our lives. But at the same time, it hasn't changed the basic nature of reality. And I think that there will be gradual steps on all of these.
mm,
The thing that has become most associated, I guess, with the idea of a singularity is this idea of an artificial intelligence that kind of exponentially grows on itself,
mm.
and I don't really see that happening overnight. It's not that I don't see it happening, but I don't see it happening overnight.
Okay, cool. Well, I'm going to ask you separately in a different section about like your talk at GF 2045. But is there anything else? Like I always ask, there might be something which has been sort of floating around in your mind that you wanted to talk about, which I haven't really thought to ask. And this interview isn't about what I think you should say. It's also about what you um, think that you should address. So, is there anything that you think would be worth bringing up that I haven't asked about?
Uh, yeah, I'll bring up one thing. I
Okay,
have to. I'll bring up one thing. Just a very brief thought.
sure.
So there's a lot of people that have been interested in cryonics
Hmm.
for a long time,
Hmm.
and they for some reason they don't do it. They don't sign up for it, and the reason I would assume is that they don't think it'll work, and I can completely understand that. Uh, the thing that makes me a little questionable, though, is people that are rich enough so that cryonics would be a trivial expense. They still might not want to do it. I wouldn't want to spend money that is something that isn't going to work. Uh, but it seems like those people would be uh, very interested in testing if it would work. And so it's been kind of surprising to me that there hasn't been a big cryonics prize out there before. Uh, it seems like the natural thing to do, or just not forget about a prize, but just somebody that is rich enough to consider these things and says, I don't know what's going on in this Alcor organization, or I don't know what's going on in any of these other organizations. I want to get to the bottom of it. Let's get who knows, some method that gets to the bottom of it. And our little prize is it's $100,000. It's 1/100th of an X prize. I tried to get Peter Diamandis to be interested in this, and I think he would have if I had brought a $10 million donor with me. Uh, but I mean, it really takes somebody of his great character to really hammer this home. Um, and it's kind of sad that nobody has stepped up to that role. So I just throw that out there. Obviously, it's a self-serving type of a thing. Although, if somebody else, if the X Prize organization tomorrow said, "Oh, well, we're going to do a brain preservation prize that's 1,000 times better than you guys, and you guys can just go play in the sand," uh, I could only say that's fantastic for humanity. Uh, and so, why isn't Paul Allen getting up to the bat and saying, "Why don't we see if this is going to work?" Why isn't uh, Elon Musk saying, you know, this space stuff is great, but I'd want to also see stuff 100 years from now. You know, is that possible? Any of these people that are doing these great things could easily make this happen. They could just make it happen themselves overnight.
Mm,
Just say, okay, here it is. Let's figure out brain preservation. You know,
mm,
they don't have to talk to me. There's plenty of scientists out there that they could just, you know,
mm,
they could drop some cash on and get the real balls rolling.
mm.
Um, and I don't see why they wouldn't do that because if I had $1 billion in my pocket, that's where I'd spend it.
It's interesting. Have you've obviously spoken to Peter Diamandis. Is there any objections he had?
No, no. I mean, I don't know what his thoughts are on this. I know that they've looked into a kind of a cryonics type of a prize, although it was for cryopreservation of organs. It's on their website. I don't know if that's going to be funded at any time. Um, uh, there's there's been discussions of this, but I haven't gotten uh, any reasons why they wouldn't pursue this. I think probably the reason is they don't think it's as concrete as possible, or they might have philosophical reservations against it. I do know some rich people that have reservations about it from a philosophical side.
Mm, fair enough. And we've probably already discussed those particular philosophical objections. Yes. Yeah. Okay. Cool. All right. So, yeah. I'll finish the interview. Oh, this part of the interview, and I really do appreciate you spending all this time. It's been a
No
good, long, and very informative, exciting interview. That's going to go up in a couple of parts. So yeah, I'm looking forward to hearing people's reactions. It's going to be out there. I'll post some links to it on Facebook and whatnot. But yeah, thanks very much for spending the time for this interview, Ken. It's amazing what you're doing, and I wish there were more of you, more people like you, and if we could clone you or something,
God
that
help
would
the
be
world.
awesome.
No, no. God help the world.
Okay, all right. Well, I'll just yeah, I'll stop the recording here. Thanks so much.
Great.